Good afternoon, I'm pleased to be here and give you a brief snapshot of some of the things that are relevant in the human health space as it relates to cancer. When Steve asked me, I kind of hesitate a little bit because we're in the midst of the preparation of a large AIF grant. So I'm the first, but I'm probably the most ill-prepared speaker that you'll hear this afternoon. I literally just finished the presentation about an hour ago. So, and actually, since all these ideas were kind of going around in my mind, actually they're going around because I don't grasp them completely. Most of what I'm going to talk about is something that others in the team are experts on. So I'm kind of the integrator or the lay person of the team trying to relate this information. So I'd like to talk a little bit about genomics as they apply to cancer. When we think about human diseases, it's probably the one that we associate as a disease of the genome. We've known ever since we've started looking at cancer that there's a vast variety. We didn't use the word genomics at the time, but even looking through a microscope, it's pretty easy to see how strange and disfigured and dysmorphic these cells are or become because of what changes have occurred at the genomic level. So be it at the cellular morphology level, when we started to be able to look at the chromosomes, we started to see very strange things, changes, deletions, fusions. And as the science developed, we started looking at cancer through the optics of genomics using gene expression profiling, and we could see that it's a complete chaos that's going on in those cells. And as the technology continues to advance, now we can use sequencing to look at individual cancer cells or cancer genomes, and again we see that things are pretty much in chaos. So we're trying to understand a disease that has no rules. It's basically almost survival of the fittest for the cancer cell, but it isn't following any set rules. So it's a huge challenge in trying to treat cancer in a very logical way, because we can say that every tumor is different and every cancer patient is different. So it can show you the degree of challenge that we have in trying to tackle this disease. Personalized medicine, we hear that terminology quite a bit, but I think it probably best applies to the field of cancer. And really the second statement is how do we move forward when we have a cancer patient in front of us in order to determine what is the most appropriate therapy for that particular patient at the stage of his disease, because if it's an early stage disease, it may be a very different course of action than if it's a late stage or a metastatic disease. So personalized medicine is something that is very well tailored or titled for how we treat cancer, or how we're going to treat cancer going forward. So the Human Genome Project, it's now 12 years since it was completed, but we're still benefiting in enormous ways of the enormous return on investment of information and technology advancement that this has brought. And obviously the Human Genome Project was a huge sequencing project. So the sequencing technology itself has moved from what it was a decade ago to something now that is really within everybody's grasp. And that's giving us better tools in how to deal with the challenge that cancer is. The new sequencing technology is really, and I was impressed, and this is about a year old, I believe, this technology. And it's really impressive in that we're really moving towards literally a fusion of biological information and data. Data in the form of electrical or semiconductor circuitry. And when our body, our cells make a DNA molecule, it's an enzymatic process where a nucleotide is added to a growing chain. So people have thought that maybe this was an innovative way to deal with sequencing. And what they exploited is they used the fact that every time we put a nucleotide on a growing chain of DNA, there's a hydrogen molecule that's liberated, or a positive charge. And that positive charge, it would normally just, drift off into the cell and not have much effect. But when you have very sensitive instruments, very high high level of technological measurement, you can measure that hydrogen molecule. So the sequencing technology that I'm going to show you here uses the fact that every time you incorporate a base, you produce a positive charge, and you measure that positive charge. You're not measuring DNA, you're not measuring a molecule per se, it's just a little positive electron or proton that you're measuring and displaying it electronically. In the instrument, and I'll show you what the instrument looks like in a moment, we get that positive charge. Every time we flood the system with a nucleotide, we know the basic code of our DNA is 4 letters, basically G, A, T, and C, which are the 4 nucleotides. So every time the instrument is flooded with a nucleotide, we get, and it gets incorporated, we get a positive signal. So that is then read by the pH, it's literally a pH meter that reads the sequence. So when we get a nucleotide that doesn't match, there's no signal, there's no proton release, so we don't get a signal. So we just replicate this experiment hundreds, thousands, millions of times, and we can sequence the entire genome using this approach. And the reason we can do it is it's done in a massively parallel way. So this simple reaction is done time and time again, and we get a measurement, and the computer software measures the proton that's emitted. This is the chip, and this is really where biology gets fused to computing power. We have these individual wells where a small strand of DNA, the one that we're analyzing, sits in, and you can only have 1 strand per well. And we flood the instrument or the chip with all the nucleotides. And every time we get a proton, we've got the ion-sensitive layer that measures the little spike in positive signal. And that gets interpreted by the instrument. So this instrument, believe it or not, the human genome took about 10 years to complete, and it cost $3 billion. We can now sequence 1 human genome in 1 day, in 8 hours actually, for less than $1,000 using this technology. So in terms of power, we've gone from something that was boutique and would be nice to have to something that everybody can have. It's almost the democratization of sequencing. So in the field of cancer, we now have the ability to receive a patient sample from a cancer patient and analyze the cancer genome in that patient. And all that is done in this cute little box that is about the size of this podium here. It's a 6-figure purchase, but not high 6-figure. It's a low 6-figure purchase. And that is the chip that is used for the analysis. It really looks like a semiconductor chip that you'd find at the core of your computer. So what is limiting this technology now is basically they can't make chips powerful enough to keep up with our ability to probe the human genome. So we're basically a victim of Moore's law when it comes to doing more with this technology. Now this instrument here it's called a personal genome machine. It came out about a year and a half ago. We can sequence all the genes with that. But the new technology that's coming out in September, and we put our name in the queue to receive one of those. It's called the ion proton. You can sequence not only every gene, but all of the DNA. So DNA that doesn't encode for genes probably makes up about 90% of the genome. You can sequence all of that in 1 day. So it's an incredibly powerful technology that is going to give individual researchers or research teams the ability to go into a cancer genome and start understanding what's going on. And the reason why we need to know what's going on is like I said, cancer doesn't follow any rules. It's basically whatever it takes to be more effective at doing what it does. That's the steps that it will employ. And I saw this, and recently it's a paper that came out last year. It really shows that cancer is an evolutionary process. Our cells, from the time that we're a fertilized egg to an adult, undergoes a certain amount of divisions and changes. And this is where we find an integration between our heredity and the environment, and how things usually go well, but sometimes in the case of cancer can go awry. So as our cells are exposed to our own heredity and environmental factors, cells will start accumulating mutations that can lead to cell growth, maybe benign tumors, and eventually to more invasive and potentially lethal tumors. So these things just tend to pile up in the cancer cell. And again, there's no rules. It does so according to whatever the cancer cell needs to overcome a situation and to survive. So some of these mutations are important, and we'll call them the driver mutations, but there's a whole series of mutations that are just passenger mutations. And some mutations will actually provoke by the fact that we're trying to kill a cancer cell. So because we do that, the cancer cell reacts and develops resistance mechanisms. So at the end, you've got something that is very different from the adult or the human cell that had a normal function. There are literally hundreds of cancer pathways that can be altered, changed, deleted, mutated, silenced, you name it. So the cancer cell, like I said, doesn't follow any rules. It does all of these changes to take advantage of different biological processes, either to avoid cell death and just keep proliferating or growing. It'll do it to avoid the normal rules of a cellular environment where cells don't invade other space. It will silence pathways or cause mutations that will allow it to overcome that. Or it will mutate systems that allow it to spread to other distant organs. So this is what a cancer does. But one thing that we've only recognized recently is when it does that, it creates vulnerabilities for the cancer cell. But we really, because we couldn't sequence the cancer genome, or we only had limited ways of exploring it, we couldn't really uncover all of these potential vulnerabilities. And really, what I'd like to talk to you about now is how do we exploit those vulnerabilities? How do we find cancer's Achilles heel? Literally beat it at its own game. And like I said, when a cancer cell decides to mutate gene X for its advantage, that is something that we should be looking to exploit. And that whole concept is one that we coin synthetic lethality. Normally, the pathway or vulnerability that we'd exploit would not have a significant impact, but because the cancer cell has already changed things, it then creates its own vulnerability. And I'll try and illustrate that through this simple figure. In a normal cell, we have different pathways that act in parallel. They may be interrelated, but they may be redundant, and we call these systems usually capacitors. So a normal cell will have two systems that allow it to respect the rules. A normal cell will follow the rules. A cancer cell will do what it wants and alter these genes to gain some selective advantage. But in doing so, it opens up that vulnerability that I mentioned. And what synthetic lethality proposes is to try and find the vulnerability pathway that the cancer cell is exposed, and hopefully use that to provoke cell death. So this is a strategy that is being used. There are currently investigational drugs that are in clinical trials that were identified using this methodology. The most famous one or known one is the inhibition of the PARP protein in cells that have lost BRCA. So this has uncovered a unique vulnerability in the cancer cells that it can be used as a therapeutic option. So this is a strategy that is part of the AIF application that I mentioned to you earlier. The strategy that we employ to uncover these pathways is quite unique, and again, it uses the power of genomics or what genomics has brought to the scientific community. We use a viral system to deliver a small piece of inhibiting RNA or nucleotides that target every single gene in the human genome. So each virus has one gene that it silences. So when we transfect a population of cells using these viruses, it will silence one gene and one gene only. So if we do that in a cancer cell, we can literally probe every single gene in the human genome to see if that the loss of that gene uncovers a vulnerability in the cancer cell. So using a screening assay, we have a positive where the cancer gene is present, and the cancer scenario where that cancer-altered gene is absent. And by treating both cell lines with the same viral inhibitor, we can uncover genes that are in combination with the cancer mutation lethal. and then we can start looking at developing a therapy that will selectively inhibit the cancer cells and have no effect on the normal cells. So to summarize, genomics has really been key in helping this field move forward. First of all, by giving us all the information that we need to be able to study these diseases, and secondly, giving us the tools like RNAi, using the viral systems to inhibit genes at the genome-wide scale. And hopefully, from this type of research, we'll be able to identify new targets for therapy, but also there are alternative screening methodologies that allow you to identify the compound directly. So hopefully this will change the way we take on cancer in the next decade. So thank you for your attention.